So, hello and welcome to the first of our webinar in this new series. Um, we're so pleased that you could join us today. So, welcome to Fighting Cancer from Within, using the body's own immune system to tackle cancer. Hopefully you're all keeping well, and we're really pleased to see you all here today. My name's Laura Chapman, and I'm the Public Involvement Manager. And I am here today to facilitate today's talk alongside our expert, Dr. Claire Pallis, who you can see on the screen. So we're really pleased to see so many of you wanting to come along and listen to this. And it's the first event in our series of easy to understand online talks about the health research that's being carried out at the Biomedical Research Centre, or the National Institute for Health and Care Research Biomedical Research Centre. Today's talk is going to focus on cancer research, but there will be many more talks covering other everyday health conditions over the next few months as well. So again, we're really hoping that you'll sign up for those. So what is the NIHR Birmingham Biomedical Research Centre? I know some of you might be wondering, and if you haven't come across it before, I've got a brief explanation for you. We're a collaboration between, or all the biomedical research centres are a collaboration between universities and the NHS. So we're managed here by the University Hospitals Birmingham NHS Foundation Trust in partnership with the University of Birmingham. So along with our other partners that you can see here on the slide. So it's slightly wider than just Birmingham, as you can see, much further afield. We are publicly funded by the National Institute for Health and Care Research. That's the NIHR acronym in our title. And we're not the only one. There are 20 biomedical research centres funded in England. And they're all funded to improve people's health. So what do we do in Birmingham? Well, in Birmingham, what we're doing, or the Birmingham Biomedical Research Centre, I should say, because you know it's wider. At our research centre, we've got doctors, nurses and other healthcare professionals working with researchers to take new scientific discoveries that are found in the science laboratories and turn them into diagnostics, such as tests or procedures, which help to identify illness and injury and treatments. So our research specifically focuses on the causes and effects and treatment of inflammation. Now, you probably know inflammation is a normal part of the healing process, and you've probably experienced it yourself. You know, if you've had a bang or something like that, you might have had some sort of inflammation. It might be that you've had, you know, a temperature or something like that that may have happened, and we know that that is part of the normal, say, healing process. But sometimes the body can overreact, and too much inflammation causes further problems. And it's linked to many health conditions, such as things like, you know, arthritis, infectious diseases, diabetes and liver and gut disease, and cancer. So that's what we're going to talk about tonight, and you're going to hear this from our expert, Claire Pallis. So before we get started, just a quick reminder that we also love to hear from you. So there's a Q&A function. Can you see in the bottom of your screen, there is a option there, and if you click on Q&A as we're going along, and you want to post a question, you can do that there, and I'll look at those. And then what we'll do at the end of the webinar, if there's lots of questions, we'll try and pick the most popular ones, or if it's just a few questions, we will ask Claire those, because we've got a little bit of time afterwards in order to hear from you and be able to speak with you like that. So just a quick reminder, we can't offer any medical advice, so please don't ask any questions about that, because we won't be able to answer them. But this webinar is being recorded, and again, we will share a copy of this with you tomorrow. So you'll have that along with any other details as well. So again, if you want to watch it again, or there's something you wanted clarified or want to go back to it, you've got that facility to do it. So I'm just going to ask you though, very quickly, something about you. So could you just have a look? I've just launched a poll, and in that poll, I'm asking to find out a little bit more about who's in the room today, so I can see some of the names. And some of you already know, which is great to see you, and some of the people I don't know. So we're just trying to get a sense of who actually is attending today, and just make sure, hopefully, that it's all pitched at the right level. So hopefully that's come up because I forgot to press. So has that come up now? So what we're asking you is, are you a member of the public, or perhaps you're an academic or a clinician that's already working in this kind of field, or perhaps you're just interested and you're already part of the university, perhaps, or a trust. Oh, that's fabulous. So thank you very much for taking time to just quickly answer those. I'll give you a few more seconds to do that before I close that. And that's super. I'm just waiting for a couple more people. Thank you for doing that. It always feels a bit odd when you can't hear people talking back, doesn't it, Claire? But what I'm going to do now then, I'm going to just keep that open, but I can see a few more people have said something. And we've got quite a number of people to get through. So you're all very adept at doing that. That's brilliant. That's super. In that case, I'm going to end the poll now, I think. And thank you all for doing that. Thank you very much again for joining us. And I'm going to switch up my camera and pass you over to Claire, and I'll see you when we come to the questions and answers. Over to you, Claire.
Thank you very much, Laura, for that introduction. And just to echo Laura's thanks for you all to join this evening. I'm really blown away by how many of you signed up to hear about our work. It's a real privilege to talk to you about it. So thank you very much. So as Laura mentioned, one of the areas of interest of the Birmingham Biomedical Research Centre is cancer, and specifically cancer inflammation. And I am part of a group working on cancer inflammation as part of the Birmingham Biomedical Research Centre. And we're trying to understand how to harness the power of existing cancer therapies, immunotherapies, that work by boosting the immune system. And in particular, my group want to try to understand why some people experience really severe side effects when treated with this type of cancer therapy. And the type of drugs that we're particularly researching are immune checkpoint inhibitors. And these are a type of immunotherapy that I'll tell you about more as we go further into the talk. So in order to try to understand why some people are experiencing side effects, we set up a research study, which is open in 40 centres around the UK. And we're recruiting patients being treated with immune checkpoint inhibitors and studying what happens. So what are their responses as they go through their therapy? And what we're hoping to develop as a result of our research are tests to predict who is at risk of these serious side effects, so we can use them alongside tests that predict how effective the therapy will be, so how much of a response there'll be in terms of treating the cancer. So cancer immunotherapy, which includes immune checkpoint inhibitors, aims to boost the existing immune response to cancer to get a better response against the cancer. And we know that these types of drugs are very effective in patients with advanced melanoma, which is a type of skin cancer, and advanced lung cancers. So prior to their discovery, only half of patients diagnosed with advanced melanoma survived to 6 months post-diagnosis. Treatment with one immune checkpoint inhibitor increased this so that half of patients were surviving now 2 to 3 years. Treatment with combinations of 2 immune checkpoint inhibitor drugs increased this even further. So as you can see, immune checkpoint inhibitors have been really revolutionary drugs for patients with advanced cancers. And we know in 2019 that over 15,000 patients in the UK alone were treated with immune checkpoint inhibitors. And since then, we know their use has increased as we've started to use these therapies to treat other types of cancer, and also as we've started to learn how to use these therapies in patients with less advanced cancers. So immune checkpoint inhibitors work by taking the brakes off the immune system to try to help immune cells, particularly T cells, recognize and kill cancer cells. However, activated T cells are not specifically activated to only recognize the cancer cells. And this can result in normal cells being destroyed as well, resulting in what we call immune-related adverse events. Now, unfortunately, these immune-related adverse events are actually quite common. So about 30% of patients treated with one immune checkpoint inhibitor will experience an immune-related adverse event. And this rises to about 70% in patients who are treated with combinations of immune checkpoint inhibitors, so 2 different immune checkpoint inhibitors. So immune-related adverse events are like the symptoms experienced by patients with autoimmune diseases. So some of the diseases Laura mentioned in the introduction, things like inflammatory bowel disease and psoriasis and others. So immune-related adverse events can affect any organ in the body, as shown here by this figure. And as I told you in the previous slide, they're commonly experienced by patients treated with immune checkpoint inhibitors. And at the moment, they're really unpredictable. We can't predict which patients will be affected. We can't predict which organs will be affected. And we also can't predict the timing. So whilst many of the milder immune-related adverse events are easy to manage, some of them are much harder. And unfortunately, about 1% of patients treated with immune checkpoint inhibitors die as a result of the immune-related adverse event that happens. And in particular, we know that immune-related adverse events that affect the lungs, causing pneumonitis, and the heart, causing myocarditis, have a particularly high risk of death. This said, when I have spoken to patients treated with immune checkpoint inhibitors who have experienced serious immune-related adverse events, they have all said that even if they knew those adverse events would happen to them, they still would have taken the immune checkpoint inhibitors. And this is because these are patients with advanced cancers and they don't have many other options. And immune checkpoint inhibitors have given them extra years of life and seeing all the amazing things that happens in their lives during that time. So they wouldn't change anything about that. And we don't want to prevent people from receiving these life-saving therapies, but we want to be able to inform patients and the clinicians of what the risk of side effects is, particularly as we start to use these therapies earlier in patients with less advanced cancers. Being able to balance the risk of side effects versus the chance of benefit from therapy is really important. But in recognition of the impact that immune checkpoint inhibitors have had on cancer patients, the scientists that discovered how to target immune checkpoints as cancer therapy received the Nobel Prize in 2018. So this was James Allison and Taksu Honyo. And what I'd like to do in the next few slides is tell you a bit more about the immune system and how immune checkpoint inhibitors have been an effective cancer therapy. So the immune system is probably best known for its response against external threats, such as bacteria, shown here, and viruses. So when the immune system recognizes some non-self molecules, it launches a coordinated attack involving many of the cell types that are shown here as part of your immune army. The immune system can also recognize cancer cells. These are harder to spot because they're more similar to our normal cells than bacteria and viruses are. But as a result of changes that happen in the DNA of cancer cells, they can start to produce unfamiliar, abnormal molecules on their cell surface. And this is recognized by members of the immune army so that they can destroy these cancer cells. If, however, cancer cells manage to evade the early immune surveillance systems and carry on undetected, they can acquire further mutations, and then that's when they cause problems as cancer cells. So cancer cells are unfortunately very skilled in evading our immune system, and they do this in a number of different ways. So some cancer cells stop producing those abnormal molecules on their cell surface or hide those away from the immune system so that the immune system can't recognize the cancer cells anymore as foreign or abnormal. Cancer cells can also secrete substances into the microenvironment around the tumor to inhibit and make it an inhospitable environment for the immune cells. And they can deplete really essential nutrients that help immune cells survive and stay active. And cancer cells also recruit other cell types to aid in their defense. So they recruit cell types that send out stop signals to the T cells, the effector T cells that want to come into the environment to kill the cancer cells. And the last thing I'm going to show you on this picture here is that cancer cells can also take advantage of inbuilt systems that control the response of immune cells so that cancer cells can turn immune cells off. And I'd like to tell you about one of these immune checkpoints in the next slide. So on this slide, we have an immune cell, a T cell here in green, and a cancer cell here in blue. And in order for this T cell to become activated, it needs to receive a signal that it recognizes as non-self, as something that it needs to mount a response against. And it receives this signal to its TCR, its T cell receptor. And this is the activating signal that turns this T cell on and makes it active. But the T cell has many other receptors on its cell surface, including one, PD-1. And this one here can receive a stop signal, which is often expressed by cancers like melanoma and lung cancer on the cell surface. So this stop signal can be sent out by the cancer cell and received by the immune cell, the PD-1 cell, so that this would dampen down the T cell's ability to recognize and kill the cancer cell. So when we give patients immune checkpoint inhibitors, what we're giving them are molecules that bind very specifically to either the receptor or to the stop signal from the cancer cell, so that these two molecules can no longer interact and the stop signal is no longer sent to the T cell. So it's only receiving the activating signals. And this allows the immune cell to recognize and to kill cancer cells. So it's boosting the immune cell response and taking the brakes off these T cells. But unfortunately, we can also be taking the brakes off T cells that recognize normal cells. And that is when we cause immune-related adverse events. And so my group is trying to understand why some people experience immune-related adverse events, and particularly very severe ones. And we predicted when we started our research that there might be differences in patients' genetic material, their DNA code, that might explain why they respond differently when they're treated with an immune checkpoint inhibitor. We also think that by studying immune-related adverse events, it might help us understand a bit more about response to therapy as well, which we know varies a lot between individuals treated with these drugs. So our plan was to recruit patients being treated with immune checkpoint inhibitors and to study their DNA to look for differences. So our cells contain around 3 billion pairs of letters, and we know that many of these letters are different between individuals. And what we're trying to do is to identify these letter changes that are more common in people who experience an immune-related adverse event. So if we consider those that experience the immune-related adverse event to be in this group here on the left-hand side, what we're trying to do is to identify letter changes indicated here by the people in red that are more common in this group compared to the group of patients that do not experience immune-related adverse event. And we hope by discovering several or multiple of these different changes that are more common in the group that experience serious immune-related adverse events, we'll be able to develop tests that we could use to predict the people who would be most likely to experience these severe events before treatment is started, so to help with that decision-making process. So the study that we set up is called ICI Genetics, and as I mentioned, it's open in 40 hospitals around the UK. And we are recruiting patients either just before they start treatment with an immune checkpoint inhibitor, or we're asking people who we know have experienced a severe grade immune-related adverse event to participate in our research. And patients are asked to give consent for a sample of their blood and for relevant sections of their medical record to be used by us in our research. And the clinical teams that we work with across those different sites share information with us about the patients who've consented to take part in our research. And this information that we collect includes the type of cancer that the patient is being treated for, the types of drugs that they are being treated with, and some information about their health at the point of starting immune checkpoint inhibitor treatment. We then also ask for the clinical team to report back at 3 months, 6 months, 12 months, and 24 months after the patient started therapy to tell us about any immune-related adverse events the patient has experienced, tell us information about how they've been managed, and information about how the patient's doing in terms of response of their cancer to therapy. And so far, we've recruited 2,635 patients, and the study will remain open until the end of this year. So we've begun to analyze some of these DNA samples from the patients recruited to our studies. And in our first analysis, we included participants who had experienced a severe immune-related adverse event. And we also included patients who had received 6 months of therapy, at least 6 months of therapy, and experienced no or only very mild manageable immune-related adverse events. So this first analysis included 566 patients. The majority of patients experienced multiple grade 3 events affecting 2 or more organs. And the organs that were the most commonly affected by immune-related adverse events were the skin and the digestive system. Using the information that was provided by the clinical teams, we calculated a toxicity score for each of our participants. And these scores ranged from 0 in patients who experienced absolutely no immune-related adverse event and increased up to 12, with the patients scoring 12 being those that experienced immune-related adverse events affecting multiple organs and of high grade, so the severe events. And what we did in our analysis was to look for changes in the DNA that were more common in those with the higher toxicity scores. And we identified 2 letter changes that were significantly more common in patients with higher toxicity scores. So now going forward in our next analyses, we are analysing more DNA samples from hundreds more patients treated with immune checkpoint inhibitors. And we want to check if those results that we found so far are the same in the extra samples. And we also hope that we'll be able to find some more promising DNA signals. Now that we've got larger sets of samples, we also hope that we can look at specific immune-related adverse events, such as those affecting the digestive system, because we do hypothesize that there might be, or do think that there might be certain changes that are only going to be able to predict risk at a particular organ. And we also hope with this expanded sample size that we'll also be able to start looking at genetic variants, DNA changes that might predict risk of some of the immune-related adverse events that we know have a very high risk of death. And they're the ones I mentioned earlier that affect the lungs and the heart. So our long-term goal from this research is to develop a test that can be used before treatment starts to predict risk of serious immune-related adverse events, so that in the future cancer therapy will be safer for patients and more tailored and more personalised to patients. So I'd just like to acknowledge the NIHR Birmingham BRC patient and public involvement and communications teams for helping me put together this talk. I'd also like to thank the NIHR Birmingham Biomedical Research Centre and Cancer Research UK for funding our work. I've worked with two fantastic PhD students during this project, Ikshin Chin and Annie Georgiou, and we're supported by many staff at Birmingham, oncologists and lab staff, and clinical staff, as well as all the staff at the other sites that recruit patients to ICI Genetics, and we're very grateful to all of those. We also have some fantastic collaborators at some of the other biomedical research centres and beyond, in both here in Birmingham, in Brighton, in Oxford, Newcastle, Leeds and London. And most importantly, I'd like to thank all the patients and the relatives who've participated in ICI Genetics. Our research wouldn't be possible without this. And I'm very happy to answer any questions that you might have. And thank you again so much for attending this talk.
Oh, thank you, Claire. That's absolutely fascinating. Really appreciate you giving a little bit of a view into what you've been doing, what you're working on. It sounds so exciting and really good news for us all. I'm going to now then open up the floor to all of you that are attending today and just say, are there any questions, or have you got any queries or anything that you'd like clarified after hearing from Claire? I know there is a lot to take in. And as I say, we know as well that you are going to have a copy of this. And again, you might think of things afterwards. And again, we can talk about how you can stay in contact with us afterwards as well in a minute. But I'm just going to have a look through and just see if anyone has asked any questions. Okay, so we're getting a nice few numbers up. I might just ask one of the questions to you, Claire, to start with then. The first one I'm going to ask you then is, why do you think that IC drugs work so well in these specific cancers, and how might they be made more effective or made effective in other cancers?
Yeah, that's a really great question. So we know that both melanoma and lung cancers contain a lot of mutations. So a lot of things that are very specific to the cancer and not shared by normal cells. And so this makes these cancer cells very visible to the immune system. And even if those cells are then secreting the molecules I mentioned about, sort of trying to dampen down the immune system, when we give an immune checkpoint inhibitor, we're able to tip the balance. So the immune cells are already there. They've been pulled in by all of these abnormal cancer-specific molecules. And by giving immune checkpoint inhibitors, we're able to boost the immune system that's already there to act against cancer cells. In other cancer types, they don't produce so many mutations. They're not so visible to the immune system. So if the immune cells aren't already there in the microenvironment, it doesn't matter if we give an immune checkpoint inhibitor. The immune cells aren't there and able to mount a response, and that's why they're not effective in some cancer types. But we are learning about ways to use immune checkpoint inhibitors alongside other drugs too. And so you might have heard in the press a lot of excitement around cancer vaccines. And we hope that by using a cancer vaccine in some of these cancer types that don't normally respond very well to immune checkpoint inhibitors, we might be able to make the cancer more visible to the immune system by giving those vaccines first, so that then the immune cells are being pulled into the cancer microenvironment. So when we give an immune checkpoint inhibitor, they're able to be much more effective because the immune cells are already there. So we really think it's down to how visible different cancers are to the immune system, as to sort of how well they respond to immune checkpoint inhibitors. But it's a great question, and we're trying to understand more about different subtypes of cancers, because we do know with relatively common cancers like breast cancer and bowel cancer that certain subtypes respond a lot better to immune checkpoint inhibitors. So as we start using them more and studying them more, we are understanding a bit more about the cancer types that are most likely to respond to immune checkpoint inhibitors.
Thanks, Claire. That's a really good question as well, wasn't it? So
Yeah.
that's exciting. I've got quite a few more questions for you. So next question is, in terms of adverse events, severity and frequency, Would you say this treatment is more promising in patient safety than traditional treatments such as chemo or radiotherapy?
Yeah, again, these are fantastic questions. So the problem is that they're very different in terms of side effects. So yeah, one of the reasons we call them immune-related adverse events rather than just chemotherapy or cancer treatment side effects is because they are so specifically down to the immune response rather than other types of damage that cancer therapy can cause to normal cells. And so we're seeing very different types of toxicity when we use these. So I don't think it's a case that they're more promising in terms of patient safety. I think by using some of the research that we're doing, we're hoping to make it safer because we'll be able to highlight those patients that really are at the highest risk. And some of that work's being done with traditional chemotherapies as well. So you might be aware that patients treated with a chemotherapy called 5-FU of fluoropyrimidine are offered a test to see if they carry a variant in a gene called DPYD, which affects how that drug is metabolized. So there are lots of work going on to try and make all sorts of cancer therapy safer by trying to understand who's at the highest risk of toxicity when they're given therapies, because we're also aware that lots of patients are given, you know, one dose to fit everybody, and that's not quite right. There'll be some of us who are receiving too high a dose and others who are being underdosed. And so that's an active area of research too, to try to personalize medicine better and personalize cancer therapy better so that we're giving the right dose to get the most effective response without getting too much toxicity to our normal cells as well. So I don't think, unfortunately, that immune checkpoint inhibitor is safer. It's got a very difficult to manage set of side effects, but a lot of work is going on to try and improve that safety.
Thank you. Yeah, we have got a question relating to that as well. I'll ask you in a minute if that's okay. But if I can ask you the next question we've had, what are the common skin and GI system symptoms?
Yeah, so with the skin, it's a lot of rash is sort of the most common symptoms there, and in the digestive system, it's often colitis, so inflammation of the large bowel, and the symptoms are often things like diarrhea. Conversely, you can also get symptoms such as constipation too, but so yeah, very uncomfortable bowel essentially, and yeah, and yeah, managed a lot with drugs like steroids to try and get on top of that.
Okay, thank you very much. I've just lost the question that I was going to ask next, which is, can epigenetics be used to reduce a patient's predisposition to to developing IRAEs when taking ICIs? Sorry, that's a
Yes.
lot of abbreviations I've just used then as well, but
No, that's fine. So yeah,
perhaps
so
you
I
could
think the
just
question.
explain those in your answer.
Yeah, absolutely. So I think the question is asking whether or not just letter changes within the DNA, but whether, so we know that there are extra marks that can get stuck onto the DNA, which affects how it works and how it's regulated. So I think the question is asking whether or not we could also study those marks to understand how a patient might be predicted to respond to immune checkpoint inhibitors. And it's, again, a fantastic question. We're not so much looking at that, but that's not to say that we couldn't start doing that in the future. So yeah, we're collecting blood samples from patients and extracting the DNA from the blood. So we're only going to learn about epigenetic marks that are there on the DNA from the blood. And it may be that it's the epigenetic marks in other cell types that are more important for immune-related adverse events, but we could certainly look whether or not those DNA modifications are important, and it's a good question. We're starting with looking at the actual letter changes, and other groups are looking at things like the actual number of different types of immune cells within the immune system too. And epigenetics is certainly a really interesting area. So looking at, because we think these epigenetic marks can be affected by all sorts of things in our environment and within our bodies. And so, yeah, again, they're a really good source for potentially looking for differences between individuals as well.
Okay, another question then is, are there any cancers that ICIs don't work well on?
Yeah, so unfortunately, there's quite a lot, and as I mentioned before, these generally other cancer types where we don't find a huge number of mutations within the cancer cells themselves, and so they don't pull in the immune cells for for us to then boost with immune checkpoint inhibitors. But some of the very hard to treat cancers, like pancreatic cancer, we're not seeing particularly good responses with immune checkpoint inhibitors, but we are trying to find other ways to use the drugs in combination with other strategies to try and make these harder to treat cancers respond to immune checkpoint inhibitors.
Okay, well, thanks. And we've been asked actually for the letter changes already found in his study. Have you identified why these might result in cancer patients having more extreme side effects?
So the letter, sorry, just make sure I understand the question properly. So the letter
Sorry.
changes that we found so far,
Yeah, or yeah, for the ones already found in
why
his
they might
study,
result.
have
Yeah.
you identified why, yeah, these might
Yeah.
result in cancer patients having more extreme side effects?
Yeah, so the ones that we found so far in our study are linked with genes that we don't understand the immune function of particularly well at the moment. So that's exciting because we might learn about things that are not really known about the immune system, but it does make it harder to immediately work out how they are having an effect. But one of them we think would be more expressed in a particular type of immune cell called neutrophils. So we think it would be through modifying the immune response. But there have been findings from a group in America where they have also found a genetic change that is associated with an increased risk of immune-related adverse events. And here the biology is a little bit clearer. So this letter change affects a cytokine, IL-7, and we know that this affects its IL-7 production in a particular immune cell, which we know is important in immune-related adverse events, but also survival and response to therapy as well. And so that finding is also helping us sort of understand a little bit more around what's happening in the immune system to mean why some people respond differently to these therapies. But it also opens up an interesting point that I didn't make too much in this talk, which is that we also know there's quite a strong link between immune-related adverse events and a good response to therapy. So we know the patients that get mild immune-related adverse events actually do very well on immune checkpoint inhibitors. This is probably because those immune-related adverse events are a sign that the cancer therapy is working, that we have managed to boost those immune cells, and we are getting a little bit of off-target side effects on the normal cells. But of course, if the immune-related adverse event is much more severe, then the patient isn't going to respond so well. And we can see that in our data too, that we can see the patients that have the very severe toxicities affecting the heart and the lungs don't do as well in terms of survival.
Sorry, I was so impressed then, I forgot to take myself off mute. But I've got a couple of questions about a couple of things I think are really interesting too. About the first one is, how do you take into account genetic and ethnicity factors to identify which groups are more at risk of certain cancers than others?
Yeah, so that's a really good question. And it's a problem both in working out who's at risk of different cancers and also in terms of how they'll respond to therapy. So unfortunately, at the moment, most of our data sets, including the ICI genetics data set, are mainly made up of white European participants. And we would really love to hear from you as well about ways that we can encourage other groups to participate. Are there particular fears that certain groups in the population have about participating in research? We'd really like to try and understand that. Our study is open in multiple hospitals that serve very diverse communities, but we're not seeing that diversity in the participation in research. So we know that's something we really want to work on in the future. So unfortunately, at the moment, our research really is, whilst it's not limited to patients that are from a white European background, the majority of our participants have that background. So we would need to conduct our research in other populations really to understand whether the findings that we find, whether they are actually useful in other populations. So it is a big problem, and it's a problem faced by the majority of genetic studies around the world at the moment. But there are a number of initiatives, some funded by the NIHR, to really try and improve this, to really diversify the the people who are represented in our research studies, because we do want it to be representative. We want these findings to be useful to populations around the world.
Yeah, okay, a good point. And I'm sorry, I can't remember, but someone's asked this question about whether or not this study was recruiting children. And
No.
if not, yeah, or have you got any plans?
No, so again, that's a really great question. No, our study is only eligible, so participants are only eligible if they're over the age of 18 and able to consent for themselves. Immune checkpoint inhibitors are being used in childhood cancers, and it would be very interesting to try and also predict immune-related adverse events there as well. The cancer types where immune checkpoint inhibitors are used in children are rare, and so trying to collect and recruit enough patients who are willing to participate is a challenge there. I think we wanted to see that we could actually find something useful in this larger population of adult cancer patients first, but that would certainly be a really interesting future direction to take this work in.
Oh, thank you. Yeah, actually, on that, I did want to know something about public involvement.
Yeah.
Someone's asked a lovely question about great. I'll read out the question to you.
Yeah.
It's great to acknowledge the patients in the cohort, but would be nice to see acknowledgement of patient partners.
Yeah.
How have they been involved in this?
Yeah, that's a great question. So before we started the study, we invited a group of patients who worked with the Birmingham Cancer Research Centre to come along and have a discussion with us about our planned protocol. And so we did have some input from patients at that stage. So they helped us design the patient information sheets that we use and some of the sort of documentation that we give to the clinics to recruit the patients. But it's certainly something we'd like to do more of, and certainly as we're now getting some results, trying to feed those back to participants within our research and to hear more of their experience of these side effects. Also through Lalit Palan, who is the chief investigator at Birmingham for our site. He's also shared some interviews with patients where he's asked them about their experience of being treated with immune checkpoint inhibitors. And, we found that really illuminating as well, hearing that patients wouldn't change. It wouldn't make them decide not to take the drug if they were given that information up front. Because like I mentioned before, there isn't too many other options, and you're trying to fight an aggressive cancer. You're going to take the therapy that's giving you the best chance of fighting that cancer. But I think if we can highlight to patients their risk, like a more personal idea of risk, rather than this sort of long list of side effects that you receive with a percentage of the population that experience those, that hopefully would make the patient and the clinician just be a bit more on the lookout for some of those side effects, so that modifications could be made early on. But also, if you're then deciding between maybe taking a combination of 2 immune checkpoint inhibitors versus 1 immune checkpoint inhibitor, knowing your risk of side effects might help when you're making those decisions.
Thanks, Claire. I mean, talking of that, this is a good question about that thing, I think. So are ICIs typically used as single agents or in combination with other therapies, e.g. Chemo?
Yeah.
And what makes a good combination?
Yeah, so it really depends on the cancer types. Again, fantastic questions. It really depends on the cancer type, but particularly in lung cancer, we are combining immune checkpoint inhibitors with chemotherapy as well. So we know that that's a very effective strategy for some cancers. In terms of whether we use 1 or whether we use 2, again, that comes down to some patient-related factors and also down to the cancer type as well. But we're seeing in our research that we're seeing lots of different combinations coming through, and the study's been running since the end of 2018, and we've seen over time that the types of therapies that are used are changing, and we're seeing new immune checkpoint inhibitors coming in as standard of care for different therapies. We're also seeing that over time, the majority of the patients at the beginning were all patients with melanoma and lung cancer, and now we've got maybe 20 or 30 different cancer types that are represented in our study. So we can see over time things are really, really changing. But yeah, to try and answer your question, it's very cancer specific. It's also patient specific as well, taking into account their health and the treatments that they've already had before as well.
Great, thank you. someone has said that they read recently about a patient who's not responding to PD-1 inhibitors or immunotherapy and been given a fecal transplant.
Mm.
Have you considered how the gut microbiome might
Yeah.
also play a part alongside DNA?
Yeah, so really, really fantastic questions. Yes, so we aren't personally studying the microbiome. It's not so much our sort of expert area, but there are other groups around the UK that are. And there's a study led by a group in Cambridge called MITRE, and they're recruiting patients and asking for those fecal samples so that they can try and study and look for both predictive markers, so markers that would be available, would be there that they could measure before a patient starts treatment, but they're also getting repeat fecal samples so that they can test over time and look for on-treatment biomarkers as well. So it's certainly an area of really huge research. We aren't actively helping with that, but yeah, really watching that space. And we're hoping that in the end, it will actually, I think it's very likely that it won't be a test that just includes markers from the DNA, from the blood DNA that are used to predict risk of toxicity and chance of benefiting from the therapy. But there'll be a multitude of other things included in that test, which may well include some measurements from the microbiome. It may well include some other cell-based measures as well.
Yeah, it's a fascinating area, isn't it?
Yeah, absolutely. Yeah.
Yeah, thank you. Do you know, will the study be open access when published? And the
Yes.
person says, thanks for an informative talk. Thank you.
Well, thank you for the feedback. But yes, it absolutely will be. So it's a requirement of studies funded by Cancer Research UK and the NIHR that they are made open access. So yes, it will be. And we also hope to make sort of more accessible versions of our paper as well, so that that we can really communicate both with the people who've participated and other interested people too. That yes, we can make our papers as accessible as possible.
Okay, thanks. Is there a check, and I might need some clarification for this, but you might understand it better than I do. Is there a test to check monitor response to ICI, for example, in lung cancer patients?
So to monitor response.
Yeah,
So, I mean, so response is really monitored by the same way it is for all cancer therapies in terms of a lot of imaging of how the cancer is responding. Can we see shrinking of the primary tumor and potentially other sites where the tumor might have moved to. So at the moment, no, there's not really any kind of on treatment tests really of response, but you might be aware of tests like circulating tumor material tests, where we can take a blood test from patients who have been treated and look to see if we can find some of the tumor specific changes in the blood. And so that is being used as a monitoring test, more in research rather than in general standard of care. But that's a really promising way, I think, in the future of monitoring response. But again, not just to immune checkpoint inhibitors, but more generally, if we can tell that we are unable any longer to find some of those cancer specific changes in the blood sample, that suggests we're able to the therapy is being effective against the cancer. If we then unfortunately after a while start to see cancer specific changes appearing again in the blood, that gives us an idea that there might be some resistance to treatment. It might be the need to change treatment, and we can do that as soon as possible rather than waiting to be able to detect something on a scan to make a change. So it might be able to give us some early warning of treatments no longer being effective.
okay, thanks. Are there any tests relating to this topic and the things just discussed? So in particular for people who take biologics, who may now be at a slightly higher risk of cancer, and how soon can this be detected, if at all?
Yes, so no, I'm not aware. There may be, but I'm not aware of tests for individuals taking biologics that would be able to predict their onward risk of other cancers or side effects. I'm afraid. Yeah, not
No,
aware of
okay,
anything.
thanks. Yeah, no, we don't know then. And I know you talked about how for instance, about the FMT work as well being done elsewhere. Someone has asked whether or not there's any relation between this and NHS Gallery? I'm not familiar with Gallery, I'm afraid, personally.
No, it does ring a bell, and
Does
I
it ring a
can't
bell?
immediately think what NHS Galleri is.
But perhaps the
Um,
person may want to perhaps submit a further
yeah, can you remind
question and
us?
just, yeah, tell us in the
Sorry
chat,
about
that's
that. I've had
fine.
a mental block with NHS
No,
Galleri.
no, not at all. I can see we're running out of time and we've got quite a few more questions.
But
So,
yeah, in terms of the comment about fecal transplants, that has been
yeah,
very interesting in that we found that when you give a fecal transplant from a patient that has responded well to checkpoint inhibitors, it can elicit a response. And yes, certainly that altering the microbiome does seem to be a really interesting way of altering response to cancers, not just within the gut. Cancers more generally being treated with immune checkpoint inhibitors. And we see that if we try and use fecal transplants for patients who haven't responded and give those to somebody else, they don't do anything at all. So it's a really interesting area of research, and it is an area of active research here at Birmingham by other groups.
okay, thank you. So, has your sort of current research looked into the drug interactions between chemotherapy and ICIs?
Yeah, so we haven't. We purposefully did include in our research patients who are receiving combinations of immune checkpoint inhibitors and chemotherapy. But what we're trying to do at the moment is to try and predict the side effects that we think are really caused by the immune checkpoint inhibitors. And we're not focusing so much on the side effects caused by other chemotherapies. We do have other studies where we're looking at the side effects of chemotherapies. And so I think when you've got combinations of drugs, it's often quite difficult to work out which side effects caused by which drug. But I think as we start to build these sets of genetic tests and markers that can predict response to chemotherapy and can predict response to immune checkpoint inhibitors, they could be used together.
Thank you. And I've got a lovely question on which to end on because I think we've had nearly every single question answered. Thank you. So this is your final test, Dr. Palace. You mentioned trying to get more diverse patients in research. Is your PPI group diverse? Wow,
So
I'm going to be definitely talking about this after you.
yes.
Yeah.
So no, it's not as diverse as we would like it to be. We're definitely aware that that's a failing of ours at the moment. And yeah, certainly going forward, we'd love to try to work out how to make that more diverse, and we may well be working with you, Laura, to try and improve that. But yeah, I agree that that's certainly a big area. If we don't get the opinions of diverse people as we're designing research, that will certainly hinder the applicability of whatever we find.
Thank you so much, Claire. And I think we're just about to run out of time, really. I just need to just kind of close down, but I can see there's a few more questions that have just arrived.
Yeah.
So I think what we'll do, we'll address those in our, we can address those, can't we, with when we give our message tomorrow,
Yeah,
giving
sure.
people this presentation
Yeah,
recording.
absolutely.
If that's okay with you, because I want you to have time to have a look at these as well and be able to answer because they sound really good. And I think everyone should hear some of the answers like they've had a chance now.
Yeah.
So I just wanted to say thank you again so much for taking our time to share that in terms which were much more understandable to me as a non-scientist and hopefully to many other people on the call today. We've had some lovely feedback as well, Claire, which you probably won't have seen. But we're really pleased that you wanted to share what you're doing with us and absolutely fascinating work. So thank you very much for that. And obviously leading on from those last questions and also from your talk, we'd say, listen, we want everyone to be involved in research. That might mean that you take part in a trial. It might mean, more than that. If you do want to take part in a trial, a clinical trial, again, we've got a great website that we can refer people to called Be Part of Research. And again, you could see on here, we've just put now the link into the chat. So you can see that if you want to click on that. And you can save that in your browser. So when you leave this call, it won't go. It'll still be in your browser. It might be that you want to be more involved and you could come and do fun stuff with me. There's a very odd picture of me there, but there's some of the people that are on the call perhaps as well today as well, when we were doing an involvement, and this is us winning a prize for our involvement as well last year at Heartlands. Again, because we do a real array of different activities to try and get as many people involved in whatever they want to be involved in, because we do need, exactly like that last question, really, we do need to make sure that we've got in our public and patient involvement groups, we know that we need to make it as diverse as possible. We need younger people, older people, all sorts of ethnicities and genders are welcome to be part of our group. And so again, we're going to put in the chat now, we've just put that in, a little bit more about how you can learn more about our Biomedical Research Centre, but also how we involve people in our work. So again, you can click on those links and join. Again, you might not want to join, you might want to just find out information. So again, just have a look and click on those, because we really want to welcome as many people as possible into what we're doing, because we need to hear from you, because what you know is something that we might not have thought of, and you can really help us do that. So thank you very much for helping to shape and show your interest. And I'm really pleased to tell you that we've been talking a little bit about this. So this is a very good segue in as well, because our next webinar, which is on 15th of June, 15th of May, I should say, again, the same time, 6 to 7, is going to be by Dr. Peter Rimmer, who's going to be talking about from gut bugs to health, how changing the microbiome can treat inflammatory bowel disease, or IBD, as some of you may know. And again, we'll give you the link for how you can join that, but we would love you to be part of that. And then after that, we've got more talks. So there's one a month, always 6 till 7, and again, always going to be recorded so you can get a copy of it as well. And each of our themes will be talking about the fantastic work they're doing. And you know, many of you are, I'm so grateful, because actually some of you are people that I've worked with in public and patient involvement, and have actually influenced this webinar ever happening in the first place. And it's a result of people, you know, working and talking with people like yourselves, that this has all come off the ground, really. So again, thanks for pushing us to do that. It's nerve-wracking, it's really fantastic opportunity, and we're always really grateful to have people shape our work, aren't we, Claire?
Yeah, absolutely. It's been a real privilege to discuss our work today, so thank you all for attending.
Thank you very much, and we'll over and out now. All the best to everybody. Bye.
Bye, thank you.
Oh, and yeah, sorry, don't forget, sorry, really importantly as well, please, please, please do leave your feedback, and we've just put a link in the chat. Please let us know what you thought, especially because it's our first one and we're learning all the time. So please do give comments and ideas for future. We'd love to hear from you. So have a lovely rest of your evening, everyone. Bye-bye.